Hello students. So this is going to be a little uh, bigger one. So I'll try to break it into two separate videos. So now we'll get into the aspects of screening and selection of recombinant uh, cell as well, recombinant vector as well as transformation uh, selection of transformants also. So success of any of the cloning experiments mainly depends upon the uh, whether we able to identify the particular recombinant vector construct as well as can we able to identify the transformants, the one which have got transferred with the transgene or the recombinant vector. Can we able to clearly identify, select, screen it and select it? Screening is nothing but identification, selection is nothing but it is the taking away that particular identified cell, that is the selection. Okay, so because given that large genomic library may contain millions of copies of sequences, so which are not rapidly distinguishable from each other by simple analytical method, so that is why the screening and selection is important. Selection, as I told you, it is just selecting the cells which have got transformed with the recombinant DNA, so which is people are doing it by using either antibiotic resistance, or they are doing it by what you call it as by complementing the particular mutations. Okay, so like that there are different methods, indirect methods are there, direct methods are there here, like that. For selection, screening means it is a procedure by which a population is of viable cells is subjected to some sort of analysis that will help in identifying the desired sequence within the organism. So there are different methods of screening: genetic selection and screening, screening using nuclear hybridization, screening by using PCR screening methods, and immunological screening. So PCR screening I will not be much touching upon. Okay, so I'll see in case if you need, if you tell me, then I'll touch upon the PCR screening more. Mainly I'll be talking about the other three methods. So this is the screening and selection map. So if you go through, you will find, okay, so there are different methods people employ, different ways they are going to go for this particular method, and they do the screening. So for during the process of selection and screening, the very important method is called replica plating. So why replica plating is required? Because sometimes when we go for selection or screening method, so whatever the transformants we get, the transformants we are going to plate into a particular media. So once we plate into a media, now during the process of selection and screening, if we are going to do directly on the same plate, there is always a good chance that sometimes this particular process will be leading to the death of the cell, or the colony is not is going to become non-viable. So in that case, so directly if we are going to screen in on the plate which we directly obtained during the process of transformation, so that will leads to complete loss of that particular colony, so where even after screening it will not help us in going any further. So therefore, generally what people do is that the master plate is the one which we directly obtained during the process of transformation. Once the transformed cells are going to grow into a plate, that plate is called as master plate. So from the master plate, people are going to develop some of the means few number of the replica plates. So these replica plates, the speciality is that they have the colonies at the same position as that of the master plate. So you can see that this particular one, the master plate in B, the first plate, which is having a red dot, that is the dot just to indicate that that is the spot from where we have to see as a for the positions of the colony. So then there is at the top A, you can see it is the stamp. So just like people do seal, the stamp sealing, so in the same way for nameplates and all, the same way here what we do is we take a stamp like stuff, so which is going to be covered with the uh, aseptic or disinfected muslin cloth, or sometimes people use velvet cloth material, which can actually adhere with the bacterial cell. Now what they do, they take that particular stamp, they put it onto the master plate, and so that wherever the colonies are there on the master plate, that colony, some of the cells of those colonies will get adhered to the velvet membrane sheet, and then that will be stamped onto the empty, not non-inoculated empty petri plates. Okay, so based on the number of plates required for the process of screening and selection, so that many number of plates are going to be stamped such a way that the mark is going to be, the position of the marking is going to be maintained. So next, when we incubate that particular plates, we call them as replica plates. So there the colonies are going to grow exactly in the same position, just like that of the master plate. So now we keep the master plate separately, and we are going to do all the processing, selection, screening, all this with the replica plate. Now get into the first method of screening, that is genetic selection and screening method. In genetic selection and screening method, people are going to use the genetic characteristics are the genetic gene or phenotype, genetic genotype of that particular organism for the process of screening. So for example, here the antibiotic selections is one of the method. For example, in PBR322, which contains gene for ampicillin resistance and tetracycline resistance. So based on the insertion of that particular the transgene or the target DNA into the ampicillin or tetracycline resistance gene, so that particular plasmid, either it is going to lose that particular antibiotic sensitization. So here you can see in this slide, there is a bacterial cell which is going to get transformed with this particular recombinant DNA. So that transformed cell is not having any plasmids, and it is not resistant to the ampicillin or tetracycline, so that is sensitive for it, means it will not grow in a media having tetracycline or ampicillin. Now once the transformation is going to be done for the recombinant DNA, okay, so then the cells, because when we produce the recombinant DNA, we will not be knowing that all the vectors have got ligated with the target DNA. Sometimes vectors can also ligate by themselves, which I discussed in the classes earlier. So here the second cell, you can see it has got the vector, not the recombinant vector, the vector which got ligated by itself. So it has both ampicillin resistance and as well as tetracycline resistance. Now the third cell is the one which is having the recombinant vector, wherein the ampicillin wherein the transformation is achieved with the recombinant DNA, so it is having the ampicillin resistance, whereas tetracycline has become sensitive. That means the gene of interest has got ligated into tetracycline resistance gene. So now these particular cells, when they are going to be plated, because when we prepare now, we get all these three are going to be there in the mixture. So when we go for transformation, when we come out with after the transformation, now this, when it is plated onto a medium, okay, having ampicillin resistance, ampicillin within it, so what will happen, all those cells which are resistant to ampicillin will grow. So we know that the cell which was not got transformed cannot grow because it is sensitive to ampicillin, whereas the cell which got transformed now with the only recombinant DNA and with only vector, okay, both these cells are going to grow on this particular medium. Now our job is to differentiate and select those cells which have got our target DNA into it. So for that, what we do, we are going to first, now we take the replica plate, prepare the replica plate. After preparing the replica plate, so we prepare the replica plate onto a medium having tetracycline in it. Okay, so what will happen, because as we know the gene of interest has got the target DNA has been inserted into tetracycline, The tetracycline sensitive, our cells are going to be tetracycline sensitive, so they will not grow in the tetracycline medium. Whereas so when we do prepare the replica plate, so we will know the position of the colonies which have been growing or which have not grown. So now in the replica plate having tetracycline in the medium, so where we have to identify those colonies which are not growing in the particular plate, because they are the colonies which are carrying the target DNA. So now that particular cells can be picked up from the master plate and can be used for further. So this is a one method wherein it is also, you can say, insertional inactivation is being used here as a screening method, or you can also say genetic characteristic, which was gain of function, you can also say. Okay, and loss of function, both are going to be here used for identification or screening. Next is here we come across with use of chromogenic substance. Other than antibiotic, we can also use chromogenic substance. So chromogenic substance is going to be used in, that is the coloring substances which can color the colony. But problem here is once the colony gets colored, so we will not be able, that means the cells are going to be dying off. So here people are going to use the very famous blue white screening, which is done with the beta galactosidase enzyme, the LACZ gene. So they use the chromogenic substance called as XGAL, that is nothing but 5-bromo 4-chloro 3-indolyl beta D galactopyronoside. So now when the enzyme acts upon this, it will remove the beta D galactose from it. So removing means leaving only X, which is going to be the blue color chromosome. For this, people use IPTG, that is isopropyl thiogalactoside, is one of the analog of galactose, which is actually acts as a inducer for this particular gene, so that will make the cell to produce more amount of galactosidase, and we are going to screen. So this is the structures of them. So I'm really sorry here. Is an the same thing I was speaking about. So overwritten has become. So here the same LACZ, the cells which are going to be used for transformation, that can be of two varieties. One, like people usually go for the host cells, is going to be LAC minus. That means they're not going to carry the gene for beta galactosidase, so that they'll not be able to produce the enzyme which can convert the XGAL into X. So these particular cells remain as white in color, whereas those particular cells which are unable to produce a beta galactosidase, they are going to become blue in color. So here this blue white will able to differentiate between the cells which have got transformed with the transgene having the beta gal intact beta galactosidase gene, and also in another method way, people also use this particular method to identify the recombinant DNA which is not carrying the intact beta galactosidase gene also. So here we come across with a little variation of blue-white screening called as alpha complementation. So this is the second approach. In this approach, people use complementation. Complementation means, so the thing which is missing in one of the component will get complemented with the others. So here what they do, they have studied extensively this beta galactosidase gene as well as the enzyme protein, and they found that there are two different components within this particular protein. That is, one is the alpha part, other one they call it as omega part. The alpha part and omega part, if they are present together, then only this particular beta galactosidase enzyme will be active and can able to perform the removal of galactoside from the XGAL. So now what they have done is, during the during the vector construction, they have constructed a vector carrying the promoter regions of the beta galactosidase or the LACZ gene, along with only the first 146 amino acids coding nucleotides of the lag Z within the vector system. So now because of this, vector is not able to produce a functional protein by itself. It can produce only the first nucleo part of the, that is alpha part of that particular nucleotide, whereas the host, the which is going to be get transformed with these type of vectors, were made such, so that these particular host cells will have the beta galactosidase omega part of the gene, and so that whenever the cells are going to get transformed with the vector carrying the alpha part of the beta gal, they'll produce alpha part, whereas the host cell DNA is going to produce the omega part. Together there will be functional beta galactosidase which is going to do the removal of beta D galactoside from that particular X gal, so that the color of the blue is going to appear. Okay, so that is called as alpha complementation. So this type of arrangement we see in case of many number of PUC plasmid vector systems. They carry this means they are going to be screened by using this alpha complementation. So you can see here, okay, the lag Z dot the lag Z, so together they can give you functional beta gal which is going to give you the blue color colonies. If there is lag Z and which is not is going to be properly oriented, then means the PUC plasmid, if it is having the gene of interest inserted into the lag Z alpha, so that will not be able to produce a functional lag Z alpha, the will not get complemented, so there will be the cells are going to be colorless, remains colorless. So that's a method people use it for screening by using alpha complementation. Next is insertional inactivation. So partially this one I explained in the earlier part itself, the ampicillin resistance, tetracycline resistance gene. So here insertional inactivation is a method in which the antibiotic resistance status is going to be used as a method here, and even people use laxed status also. So here the gene of interest will be inserted into these marker genes. I use a word called marker genes, which is used for selection. So they are going to be inserted into these particular genes, and due to insertion into these genes becomes inactivated or non-functional. So this inactivation is being used for screening. So these people have seen in case of PUC plasmid, M13 plasmids where Laxid is being used, and large number of other type of derived plasmid vector systems like PBR 322 and all people have using antibiotic resistance. So even people are using another kind of feature also in insertional inactivation, that is plaque morphology. Plaque morphology means whenever a bacteriophage infects the bacterial culture lawn in a petri plate, so the places where exactly the bacteria, the cells means the active bacteriophages, wherever they are going to infect, so there they are going to create an plaque. The empty means they kill all those bacteria and only media will appear there. So this plaque morphology people are using it here. So in case of, for example, in case of lambda GT10, one of the replacement vector of lambda, so it contains a gene called CI gene. So this gene encodes a protein called CI repressor, which is responsible for the formation of lysogens. So lysogeny, you know, lytic and lysogenies. Lytic is nothing but the phase in which the cells are going to be killed. Lysogeny is a phase in which the cells are not going to be killed. So now if you find any kind of cells remains within the plaque portion, that means those particular cells are in lysogenic form. So lysogeny is active means the CI is active. CI is active means, in other words, you can say the lambda GT10 is there inside those particular cells. So this is one more method people use it for the screening. Okay, so this is called the plaque morphology. Means instead of getting completely clear plaques, we will get opaque plaques. Means plaques having few number of uh, cells grown there into a small dot colonies in that particular regions. Okay, so this is another method. Then we come back with complementation of defined mutation that I spoke about, the alpha complementation. Here is a complementation of defined mutations. Specifically, we can talk about the nutritional components. Okay, for example, it can be for an enzyme which is going to be part of a particular synthetic pathway. Let's say, or the utilization of a particular carbon source within the medium, or production of some amino acids or some nucleotides. Okay, so those particular components can be means the genes can be used as a uh, means markers for selections here. So if you are going to find an inactivation of those genes, that forms those particular cells are called as auxotrophs. Okay, the genes of the, for example, tryptophan. So one of the gene of the tryptophan operon, if it is going to be mutated, so then we can select the mutant based on the tryptophan. So if you provide the tryptophan in the media, the cells will grow. Now if they are not going to provide, they will not grow because they lack the capacity to synthesize the tryptophan amino acid. So like that, people go for screening by using number of genes. Here just given you the list, like ampicillin, hygromycin, kanamycin, neomycin, streptomycin, tetracycline. These are some of the antibiotic genes which are being used for insertional activation or also as genotype screening part. Okay, so I'm going little faster because this is a very big chapter, means concept, so where I want to touch upon as maximum as possible. So other kind of genetic method which we are going to come across is screening and selection by using some sophisticated components, such as in case of lambda GT10. Okay, so people go for this high lysogen frequency in case of E. coli as one of the method, and such strains are called as HFL strains, high frequency of lysogens. So that is that particular characteristics people are using it for screening. And even there is one more called the spy phenotype, which is based on the red and gam gene in case of lambda phages. So that's also one of the method people are employing it in case of screening. Please go through it. If you come across with any doubts, you can ask me. Now I'll okay, this I'll finish, sorry, now I'll stop. I'll continue from the hybridization methods in the next set of slides.